This session is dedicated to our discussion about the process of transcription. Um, we are going to look at how transcription happens in both prokaryotic and eukaryotic systems. So, before we dive into our discussion about transcription in bacteria and in eukaryotic organisms, let's first take a look at the process, examine the general features and elements that are involved. So we define transcription as the process of RNA synthesis from DNA strands that serve as template molecules such that genetic information encoded in the DNA is transferred to RNA. You know. Essentially, the process of transcription has a lot of similarities with replication. So both of them uses DNA as their template molecule. Both processes require the formation of phosphodiester bonds, and also in terms of the direction, okay, both synthesize molecules from 5 prime to 3 prime direction. So it's always the 3 prime end of the molecule that serves as the active site for the addition of an incoming substrate. Okay. Aside from similarities, there are outstanding differences that we also have to remember. So in terms of the template, the nature of the template is actually different. So in replication, both DNA strands are used as template as parental molecules, while for transcription, only a single strand of the DNA is utilized as a as a template for RNA synthesis. For the substrate, mostly for replication, we're talking about DNTPs, no? While for transcription, since RNA molecule is being synthesized, we're not really using DNTPs as our primary substrate. Primer is also not needed for the case of transcription, unlike in replication. The enzymes that are serving as the primary, or the enzyme serving as the primary synthetic molecule, is also different. In replication, we've been introduced to DNA polymerases. For the case of transcription, we have what we call RNA polymerases as the primary synthetic enzyme. The products are also differing between the two processes. Replication produces double stranded DNA, while transcription produces single stranded RNA that may come into different types or that may exist into different forms. So you have your ribosomal RNA, your transfer RNA, and your messenger RNA. Later at the end of our lecture, we will talk about post transcriptional modifications or edits that happen in these RNA outputs, so in these RNA types. Okay, and then lastly, of course, a very obvious difference is in base pairing. So in DNA replication, of course, adenine base A base pairs with D, G still pairs with C, while for transcription, anything that pairs with or I mean, for the case of adenine, of course, the RNA transcript will produce or will pair a U, no, to it, and then we also have our the same pairing for G and C and D and A, no. Okay. So, enough dun sa, ano, no, sa discussion natin sa similarities and differences and the general process. Now, let's take a look at the elements that are interplaying for RNA transcription to be happening all the time in prokaryotes and eukaryotes. So let's examine the template and the enzymes. It's the whole genome of or the whole all DNA that is required to be replicated, as we remember from our discussion last week. Okay, it's the whole DNA molecule, the whole genome is replicated. But when it comes to information transfer, only a small portion of that genome is actually transcribed in response to signals such as cell division, physiological changes, environmental changes that the living system needs to adapt in order to survive. No? So only a very small portion of the whole genome is actually transcripted, no, for information to be processed. And these regions of DNA that can be transcribed to become RNA transcripts are called structural genes. Okay, so the essentially when you encounter something that is labeled as a gene, therefore that's a portion of the DNA that is transcripted to RNA to allow further information flow, especially from the DNA molecule going towards the production of an RNA transcript corresponding that DNA molecule, that gene, then eventually for protein synthesis. Okay, the template strand. When we talk about transcription, now there are two things that we have to remember: the template strand and the coding strand. So the template strand is the strand from which RNA is actually transcribed, and it is also called the antisense strand. Right? The coding strand, also known as the sense strand, is the strand whose base sequence specifies the amino acid sequence of the encoded protein. Okay, so two different strands that we have to remember: the template and the coding. So let's examine them with an illustration. So here you can see a stretch of double-stranded DNA, one identified as coding and another identified as the template strand. So as we've mentioned, the template strand serves as the antisense, where the RNA is actually going to be synthesized. So your RNA after transcription will be equivalent, no, or will be complementary doon sa template strand mo, sa antisense strand mo. Okay? And your RNA transcript, of course, will be similar with your coding strand, with your sense strand. So the amino acid residues or the codons that can be identified here will be defining the sequence of amino acids in the fully translated protein. Okay. Now, we have to be reminded that for the case of double-stranded DNA, the process of RNA transcription actually undergoes, I don't know, or follows both directions. Okay, so only the template strand is utilized for transcription, and in the coding strand is actually not. No, but we have to remember, since double-stranded siya, both strands can actually be utilized as templates depending nga lang dun sa direction. Kasi remember, anti-parallel yung double-stranded natin, no? The double strands of DNA. Therefore, the segment, a particular gene or structural gene, actually possesses a coding and a non-coding sequence. So, depende sa direction, both strands can serve as templates for RNA transcription. Okay, and this phenomenon is called asymmetric transcription. So just to have a good view of how what this feature actually means, let's have here another illustration. So we have double-stranded DNA. So let's say, for example, we have two identified structural genes here. Sorry, right? And then this part here is serving as a template strand. If the direction of transcription is from this point to this point. So kung pag ganito yung direction, this serves as the template strand. Okay? While this serves as a coding strand. Now, if the direction is, of course, the opposite way, yung kabilang molecule or the upper strand will serve as the template, while the lower serves as the coding. Right? So in essence, for organisms, not only in eukaryotes and prokaryotes, even in viruses, the same process, no, is expected to be found. As long as, of course, we're talking about double-stranded DNA as template for RNA transcription. Okay. Now, so the template, we're done. Next element, the enzyme, RNA polymerase. So RNA polymerase is the enzyme responsible for RNA synthesis following a DNA dependent fashion. So you can further qualify that RNA polymerase as a DNA-dependent RNA polymerase. For the case of prokaryotes, the RNA polymerase is a multiple subunit protein at about 480 kilodaltons in size. While for eukaryotes, the RNA polymerases are also multiple subunit proteins, but they are identified to be known as, or they are identified to be three separate enzymes, so three different RNA polymerases for eukaryotic organisms. Okay, let's examine first the RNA polymerase of bacteria. So RNA pol of E. coli. Okay, so for E. coli, the holo enzyme of the E. coli, the holo enzyme RNA polymerase of E. coli is actually consisting of five different subunits: an alpha 2 subunit, a beta prime subunit, an omega subunit, and a what do you call this? A sigma subunit. Okay, so just to show you some illustration. So this is the core enzyme. So the alpha, okay, to alpha, beta prime, an omega, and a sigma. Okay, so the addition of the sigma will create a holoenzyme, while the core, of course, kung wala pa si sigma subunit, is just the catalytic unit, but not necessarily considered as the holoenzyme. Okay. Now, more information about the subunits of the RNA polymerase of bacteria. Let's examine here this table. So let's take a look at the major subunits. So for the alpha, it's about 36,512 for its weight. The function is to determine the DNA to be transcribed. So it's alpha subunit, recognizing the DNA segment to be transcripted. The beta catalyzes polymerization. The beta prime is responsible for binding and opening the DNA template, and the sigma unit is responsible for recognizing the promoter sequence for the initiation of RNA synthesis. Okay, so the different functions for each subunit. So we can really think of the subunits as as separate molecules carrying out specific functions, and they're joined together to carry out RNA transcription for bacterial organisms. Okay. Now, some side information. The molecule or the substance rifampicin is a therapeutic drug for tuberculosis, and it can bind specifically to the beta subunit of RNA polymerase, and it can inhibit RNA synthesis. So rifampicin, the mechanism of action in counteracting Mycobacterium tuberculosis is to bind to the beta subunit of the bacterial RNA polymerase, therefore inhibiting its synthesis capacity. Okay. Now, in most prokaryotic systems, we expect that the structure of the RNA polymerase, the multiple subunits found in E. coli bacteria, is the same. So, almost the same. We don't have much differences to note. No? Now, let's talk about the enzyme in eukaryotic organisms. So, for eukaryotic organisms, the RNA polymerases are labeled with Roman numerals 1, 2, and 3. And they have different products. Okay, so RNA polymerase 1 synthesizes ribosomal RNA or produces produces ribosomal RNA transcripts, while RNA polymerase 2 responsible for producing heterogeneous nuclear or heteronuclear RNA. It's your hnRNA is what you call your pre-messenger RNA. So RNA pol 2 is responsible in producing that, while our RNA pol 3 is responsible for producing other types of RNA, like the small subunit of ribosomal RNA. Our transfer RNAs are synthesized by RNA pol 3, and our small nuclear RNAs are also synthesized by pol 3. Now, side information, okay, we have this molecule amanitin, which is a specific inhibitor of the eukaryotic RNA pol. amanitin from the poisonous fungi Amanita. So it is the mechanism of action of amanitin is targeting the polymerase enzyme of eukaryotes. Okay, so two elements that of transcription that were very clear as of now is the enzyme, of course, and the template. Okay, now let's examine other elements, other important things to consider before we actually go about the process, no? The process of transcription divided into three, yung initiation, elongation, and terminations. Before we talk about those three stages, another important thing that we have to review is um, ano, no? Important sequences that serve as recognition, no, recognition sites, no, or you would remember in our discussion about DNA replication, we have this origins of replication, diba? For prokaryotic and eukaryotic systems, and in transcription, the same case, we also expect the same elements. Okay, so each region, each transcriptable region, which may be composed of multiple structural genes, is called an operon. So you probably heard of the term operon in your genetics. No? You remember the lac operon, important for lactose digestion or lactose metabolism in bacteria. Okay, so each operon is consisting of multiple structural genes and upstream regulatory sequences or regulatory regions. So these regulatory regions are not are not essentially structural genes, but they are important for the promotion of the process. No, they're important for the initiation of the transcription process. Okay, the promoter is a DNA sequence that the RNA polymerase can bind. So, and this promoter is a upstream regulatory sequence. It's key to the control of the transcription process. So, just for us to visualize, we have this nice illustration here. So, this is a double-stranded DNA. If this pink region here is a structural gene, and this cyan-colored region here are the upstream regulatory sequences. So, nandito yung promoter site. And this is where RNA polymerase binds. In prokaryotes, we have designated regions that are actually conserved universally across different species of microbes, of bacteria. No? And these regions are what we call the -35 region and the -10 region. So the -35 region, Tatagaka, and the -10 region, which is rich in adenine and thymine, no? The Tata-Ata, which we also call the Pribno box. Yeah, so these are the prokaryotic promoter sites. So they are upstream doon sa ating structural gene portion, right? So they're not within the structural gene component. Okay, so where your RNA polymerase binds. So in prokaryotes, RNA polymerase directly binds to the promoter sequences, unlike in eukaryotes. So eukaryotes are totally having a totally different story. We will talk about them in a little while. So this -35 region and -10 region are considered consensus sequences. So in an examination of multiple structural genes, okay, in prokaryotic organisms, it has been found that the -35 region and the -10 region are actually universally conserved sequences. No? So ano sila, talagang constant sila in multiple genes and also in across species of prokaryotes. The -35 region in particular, identified as the Tatagaka sequence, it serves as the recognition site and binding site of RNA polymerase enzyme. While the -10 region, known as the Tata-Ata, the Pribno box, is the region at which a stable duplex or complex of DNA and RNA polymerase is already formed. So, at Tatagaka, initial recognition. Then, the RNA polymerase bound to the DNA will slide down. Ano siya, no? Magta-travel siya from the -35 going to the -10 and then eventually the start of the structural gene. So, the -10 region is where there is stability formed, no? Between the DNA and the RNA polymerase complex, right? So now I think we're ready to examine the transcription process. Let's take a look at the general processes, the general concept. So we divide the process of transcription in prokaryotic systems into three. So there is an initiation stage, an elongation stage, and a termination stage. The prokaryotic RNA polymerase can bind to the DNA template directly, as I've already mentioned, during the initiation part of transcription. While for eukaryotes, RNA polymerase will require cofactors to bind to the DNA template together. To bind them together, meron tayong additional molecules that are needed. We will discuss them in a little while. We'll first examine the three steps in prokaryotic organisms. So for initiation, RNA polymerase recognizes the promoter sequence and will essentially start the transcription. Elongation is defined as the continuous polymerization of RNA strand, while termination is when the RNA pol stops the synthesis process and the growing RNA is now separated from the DNA template. So let's examine each stage closely. Initiation, so we have RNA polymerase recognizing the tata-GACA, the -35, and slides down to the tata-ATA, then opens the DNA duplex. This unwound region is about 17 plus minus 1 base pairs. So ito yan, no? This unwound region is about 17 to 18 base pairs. Okay. During the initiation, we can expect that the first nucleotide on the RNA transcript is always a purine triphosphate. And usually GTP is more frequent than ATP. Okay? So that first nucleotide structure remains on the RNA transcript until the RNA is fully synthesized. Okay, so this is what we call the transcription initiation complex. So the three important molecules that serve as the components of the TIC are the RNA polymerase holoenzyme, the DNA template, and of course the first nucleotide on the RNA transcript. Now, unlike DNA replication, RNA synthesis, RNA production does not require a primer. Okay. Now, we expect eventually that the sigma subunit will fall off from the RNA polymerase once that the first 3', 5' phosphodiester bond is actually established. No? Now, the core catalytic enzyme that will continue to move along the DNA template entering the stage of elongation. So, pasok na siya ng elongation. Okay, so during elongation, no, the release of the sigma subunit causes the change in the enzyme, the core enzyme. The core enzyme will continue running down the DNA template towards the 3' end, synthesizing, elongating the nascent RNA. Okay, so free NTPs, which serve as substrate, are added sequentially to the 3' hydroxyl end of the growing RNA strand. And essentially, the addition process will equate to a release of two phosphate groups. No? Magkakaroon tayo syempre ng dephosphorylation ng ating NTP as the nitrogenous base containing component will be added to the growing RNA. Okay? Now, okay, so another term that we have to be reminded, the transcription bubble. So the transcription bubble is composed of course, the enzyme, the core enzyme RNA pol, a DNA segment of about 40 nucleotides, and the growing RNA. So it's what we call, this combination or complex of molecules is what we call the transcription bubble. Okay, the 3' segment of the RNA is expected to hybridize with the DNA template, while the 5' end actually sort of overhangs. Parang naka-peek out siya dun sa transcription bubble, nakalabas yung 5' end. It extends out of the transcription bubble as the synthesis is actually progressing. So to put things together in one picture, I have this very nice illustration. So you have your RNA pol bound to your DNA duplex, your DNA double strand, and then here is your RNA strand that is growing. The 5' end, yan, nakaano siya, nag-overhang yan sa labas ng RNA polymerase mo. And your 3' is still complementary and hybridizing with your template strand. Okay, so here you can see an RNA-DNA hybrid of about 8 base pairs. And the active site is always the 3' end because it's where incoming NTPs will be added. Okay? All right, so more information or some additional stuff, mostly illustrations na lang. So let's take a look at the 3D rendering of the RNA polymerase of E. coli. So here you can see the RNA polymerase with and without the DNA molecule. So here, also another illustration. So positive supercoils are found at the at portions of the DNA that are yet to be transcripted. So positive supercoiling yan. Okay. So these are nice illustrations. You can go back to this. Again, just a review, no? Review natin ulit. So our holoenzyme will recognize -35 and -10. Then at minus 35, sliding will occur to -10, opening the complex. Then eventually the open complex will allow transcription initiation to proceed. Once that the transcription bubble has cleared the promoter, no, na recognize na okay, this is a structural gene. I need to transcribe it. The sigma subunit falls off, and elongation will proceed. Okay. An important feature of transcription in prokaryotic organisms is it's actually simultaneous with translation. So you'll remember that in prokaryotes, there is no nucleus. So there is no separation of the DNA molecule is actually accessible to the replication proteins and the transcription proteins at the same time. Translation proteins and transcription proteins are capable of accessing the necessary elements at the same time. There's no spatial separation. So DNA strand can be transcripted, and that newly transcripted strand can undergo translation also at the same time. So there is simultaneous transcription and translation happening in bacteria. Unlike in eukaryotes, where transcription only occurs in nucleus and translation happens, of course, in the cytoplasm. Okay? Now, termination. Yan. So, last stage na ng transcription in prokaryotic organisms. So, during transcription, the RNA polymerase stops moving through the DNA template. So, it's now done with transcribing the structural gene component. The RNA transcript will fall off from the transcription complex, and this termination process can actually be dependent to a certain protein, or can be independent. So yung tinatawag natin a raw dependent mechanism and raw independent mechanism. Let's first examine this raw protein. So this raw protein, when present, of course, the mechanism is called raw dependent mechanism. It's a hexamer of proteins that actually functions as an ATPase and a helicase. ATPase and a helicase. So it's capable of dephosphorylating ATP. So this raw protein here serves as a core molecule that can a core molecule where the RNA transcript can envelope itself onto, no? So para siyang yung sa nucleosome, kung maaalala ninyo. So it sort of ang function ng raw protein or ng raw factor is to pull out the RNA transcript from the transcription bubble. So ganun siya pagka raw dependent. So if it's raw dependent termination, the raw factor, the raw protein, which acts as an ATPase and helicase at the same time, is going to pull out the RNA transcript from the transcriptional bubble. Okay. Now we also have another mechanism, the raw independent, which actually depends on inherent sequences. No, sequences which we call termination sequences or termination signals, a stretch of about 30 to 40 nucleotides on the RNA transcript, usually consisting of multiple GC followed by a series of U. No, so because of this kind of signal or this nature of the termination signal, the sequence specificity of the nascent RNA will form particular stem loop structures that will terminate transcription. So here, for example, for the case of this specific protein, you can see the red parts will once after transcription, no, can produce stem loop structures. You can see here, okay, and this stem loop structures will inhibit the continued movement of the complete transcription bubble. So magsiserve siya as ano to, no? Stem loop or tawag ba natin? Parang we also call this hairpin loops, no? So here, yeah. So you can see here na once that this portions or once the stem loop structures form within the RNA transcript, that will serve as a raw independent signal that will tell the transcriptional bubble na it's time to stop. It's time to terminate the process. Okay? What else? So, yeah. How does the stem loop structure actually disrupt the conformation of the transcriptional bubble? So the stem loop structure alters the conformation of RNA polymerase, which will lead to pausing of the movement of the enzyme. Then a competition of the RNA-RNA hybrid and the DNA-DNA hybrid will reduce the stability of the DNA-RNA hybrid na kung saan nagkakaroon talaga ng transcription. And that will cause the transcription complex to be dissociated. Okay? Usually, among the base pairings, the most unstable one is U in the RNA molecule and A in the DNA molecule. Okay? Right, so we're done with transcription in bacteria. Now, let's examine transcription in eukaryotic organisms. So let's take a look at each of the stages. First is initiation. Transcription initiation in eukaryotic systems need promoter in the same respect that prokaryotes need promoters, and also these promoters and other regulatory sequences upstream the structural gene. Now, we have this what we call cis-acting elements that are specific sequences on the DNA template that regulate the process of transcription for operons or for single genes. So these cis-acting elements, they are homologous. Okay, these cis-acting elements are homologous with the promoter sequences of prokaryotic systems. Okay, itong mga ano na to, no, mga cis-acting elements na to, like the Tata box, the Hogness box. We also have the Kaat box and the GC box. So these are what we call cis-acting elements. They are found in the upstream, untranscripted or non-transcripted region of the eukaryotic DNA, the eukaryotic operons. Okay, and yun nga, no, they serve as binding elements. They can serve as binding elements for initiation to be important. Let's examine particularly the Tata box, kasi the Tata box is the one that we equate with the Pribno box of the prokaryotic DNA. So the Hogness box, yan, here, also is a consensus sequence, very rich in A and T. Okay, and essentially, in terms of base frequencies, yan, makikita natin how in multiple genes of the same organism, the Tata box is actually a conserved sequence. And eventually can be processed to find a consensus sequence. Okay, so upstream siya. Okay. So, transcriptional factors that are actually not found in prokaryotes are present in eukaryotic systems. So, unlike bacteria, unlike in bacteria, the RNA polymerase in eukaryotes does not directly bind to the DNA, does not directly bind to the promoter sequences, to the regulatory sequences. RNA polymerase 2, particularly for eukaryotes, needs to associate with a number of transcriptional factors, with about 6 transcriptional factors. So these transcriptional factors, acting as trans-acting factors, are responsible for the recognition and binding, okay, to the cis-acting elements, such that initiation will take place. Okay, so a number of transcriptional factors are important to form the pre-initiation complex. And they are outlined here. No? So we have our TATA binding protein of the transcriptional factor 2D binding to the TATA box, the TF2A and TF2B binding to the TF2D, the TF2F RNA polymerase complex binding to the TF2B, the TF2F and TF2E opening the double-stranded DNA, primarily as helicase ATPase proteins, and the TF2H, which will be added lastly to form to essentially complete the pre-initiation complex. So it looks like this. Yeah. Okay. Ayan. So last is the TF2H. I won't be particular with this because I think there are a number of I won't be particular dun sa sa concept ng trans-acting factors uh, uh, interplaying with cis-acting elements of the eukaryotic DNA. I won't be particular with that kasi marami na rin namang findings and there could be newer stuff. And I haven't, to be honest, I haven't read about more on the eukaryotic organism, on the eukaryotic transcription process. So I won't be particular with that. No? Hindi ko siya ipapamemorize sa inyo. Okay? So once that your pre-initiation complex is now completed, okay, or what signals the completion of your pre-initiation complex is the activity of the TF2H trans-acting factor, which serves as a protein kinase. It phosphorylates the C-terminal domain of the RNA polymerase, and once that it's phosphorylated, and that's the time that it can start the elongation part. No? You can elongate the RNA transcript. Now, we expect that as the complex of RNA polymerase, the complex of RNA polymerase and the trans-acting factors, as it travels through the length of the DNA molecule, most of the trans-acting factors will fall off during the elongation phase. No? So, similar naman din ang elongation ng eukaryotic systems with prokaryotic systems. Okay? Transcription and translation, though, however, do not take place in the same space. Okay? So, remember, there is the nuclear envelope that separates the DNA, RNA transcription from translation of proteins in the cytoplasm. Okay? So, just to have a good illustration. So RNA pol, as it moves through the double-stranded DNA, will of course move will of course have to factor the proteins of the histone proteins, the nucleosome core proteins. Right? Now, termination. Yan. For the eukaryotic systems, the termination sequence is rich in A and T, followed by multiple G and T repeats. No? The termination is closely related to the process of post-transcriptional modification. So our last topic for this recorded lecture are post-transcriptional modifications. So post-transcriptional modification, from the term itself, describes events, molecular events, that modify or edit the final molecular makeup or the final molecular structure of RNA outputs. We have post-transcriptional edits for ribosomal RNA, for transfer RNA, and for messenger RNA, which we will discuss in this last part of the recorded lecture. Okay? So, let's first examine modifications, post-transcriptional modifications in the messenger RNA. Yan. So the growing RNA, we call that the nascent RNA, we call that the primary transcript. And that primary transcript needs to be modified so we can have our working tRNAs, rRNAs, and mRNAs. And for eukaryotes, very, very important ang post-transcriptional modification. Okay? Because if you would remember from your genetics, probably, you know that not all parts of an operon, of a structural gene, is actually coding. Not all parts is coding, or not all parts are expressed. So, remind ko na kayo on the concepts of exons and introns, which we will discuss in a little while. So, very important that the non-expressed components of the structural genes are actually spliced off or are removed from the final transcript. No? So before we examine further, yun nga, for eukaryotes, the process of transcription and post-transcriptional edits are happening within the nucleus. Okay, so nangyayari siya sa nucleus. And the final output, the final RNA molecule, will now be exported to the cytoplasm. So, essentially, nagkaroon ng lahat ng transcription-related and post-transcription-related happenings. All of those are confined within the nucleus, within the nuclear space, the nucleoplasm. Okay? So, let's first examine modifications of the mRNA. So remember, the primary transcripts of mRNA, we call that the heteronuclear or heterogeneous nuclear RNA, hnRNA. So hnRNA are larger, longer than mature mRNA by multiple folds. And some of the modifications that are happening in hnRNA before it becomes a final messenger RNA are identified here. No? So capping, tailing, splicing, and RNA addition. Let's take a look at each of these post-transcriptional processes. Capping at the 5' end. So capping at the 5' end is defined as the addition of a 7-methylguanosine cap at the 5' end of the RNA transcript. So this cap serves as a protective cover such that RNases will not degrade the newly synthesized transcript. Okay, so it's found at the 5' end, this methylated guanosine. No? So here, so ito siya. This methylated guanosine here serves as a cap, a protective cap at the 5' end. Now, how is it added? There is a removal of phosphate groups doon sa initial RNA base, no, or the first base of the RNA transcript, and then a formation of a triphosphate, usually with the action of GTP, okay, and then the methylation of at G7, methylation at G7. Then eventually, another two things that are happening are methylation of the first and the second nucleotides after the G cap. Magkakaroon din ng methylation yung dalawang magkasunod na nucleotides after it. Okay, so that's how we describe the methyl cap. Okay, so there is your 7-methylguanosine, and then the two succeeding nucleotides will have methylations at their C2. Okay, so this 5' cap will protect the RNA transcript, okay? And it's recognized by the cap binding protein during translation. And prior to splicing, prior to the removal of the non-expressed regions of the structural DNA, the structural gene rather, nangyayari ang capping. Now, Another process is adenylation, poly-A adenylation, a poly-A tailing. So there is no poly T, no, sequences on the DNA template. So hindi siya actually based sa DNA template, the poly-A tail. This tail is separately added to the RNA transcript. And similar with capping, it's a process that is done prior to splicing. And also it takes place within the nucleus. So don't have a nice illustration here now. But essentially with poly-A tailing, I think let's go back to ayan. So, okay. So, here, the cap, yan, and then the poly-A tail. So, isipin nyo na lang, ano siya, no? Sandwich between the poly-A tail and the 5 prime, the 5 methyl cap, no? The 5 prime 7 methyl guanosine cap. Sandwich between these two elements, these two post-transcriptional edits, is the RNA transcript. Okay? Next, okay, then mRNA splicing. Okay, so the matured mRNAs, as I've already discussed, are shorter than your your DNA template, shorter than the heteronuclear RNA precursor molecule. Okay, we have this concept of split gene, where the structural genes are actually composed of coding and non-coding regions that are alternatively separated. So let's say we have this structural gene here. The ones represented by letters A to G are non-coding regions, while the ones represented by numbers 1 to 7 are coding regions. So separate sila na coding, non-coding, coding, non-coding, coding, non-coding, coding. So, gano'n, no? Ito yung concept of split gene. So, we have your exons and introns. So, our exons are the coding sequences that appear on split genes and primary transcripts. And they will be expressed as matured RNA. While introns are the non-coding components, non-coding sequences that are transcripted still into primary mRNA, but they need to be cleaved out of the final product. No? Kasi they do not contribute anything to the final molecule. So a process of mRNA splicing has to be carried out in order to remove all the non-coding sequences. And yeah, no, going back, both exons and introns are transcripted from the DNA template, but only exons will be kept. Only the coding sequences will be kept in the mature RNA. The introns need to go out. They need to be thrown out of the window. So they have to be spliced out. Okay? So how is splicing taking place? So the splicing mechanism is actually carried out by, or it is identified as twice transesterification processes or phosphoryl reactions. Okay, so let's say, for example, we have our split gene here composed of exon 1 and exon 2. In between them, we have an intron. This intron here needs to be removed. No? A nucleophilic attack at the 5' end of the intron will take place. So that's an initial phosphoryl reaction. Then afterwards, a lariat will form. Okay, this structure here is called a lariat. This is a unique structure. And then the second phosphoryl reaction will be a reaction against the 3' end of the intron. So the intron will totally be spliced out of the molecule of the structural gene. And then there is going to be ligation of the 3' end of exon 1 here and the 5' end of exon 2. Then the intron is just digested because it's not needed. Okay. The whole process of lariat formation, the excision of an intron in between exons is carried out by the spliceosome, or the protein complex carrying out the phosphoryl reactions, the twice transesterification processes. So here, let's just examine. So here you have your first transesterification attacking this end, I don't know, the 5 prime end. Okay, then so the 5 prime end will be, I don't know, will be released and makakat out siya. And then the second trans esterification will essentially release the intron from the between the two exons, and the two exons will be ligated afterwards. Okay. Now the last post-transcriptional edit happening in mRNA is the process of mRNA editing, which takes place at the transcriptional level. One gene can be responsible for more than one protein, for multiple proteins. No? So the importance of mRNA editing is that gene sequences after post-transcriptional modification can serve as important sources of information for multiple proteins, no? for multiple differentiations. So for example, the APOB gene in humans, it's composed of a heteronuclear RNA that is about 14,000 bases long. In the liver, this gene, the APOB100 gene, is about 500 kilodaltons. But in the intestines, it's I don't know, it's less weightier, or it weighs less, about 240 kilodaltons, and it has a different conformation. And the change is from CAA to UAA at position 6,666. So at that position, yung difference na yon, nagkaroon ka ng dalawang magkaibang protein but coming from the same RNA primary transcript. Okay, that's mRNA editing, right? So done. Done na tayo sa post-transcriptional modifications for mRNA. Now let's take a look at post-transcript edits in tRNA. So what modifications are existing? So similar then with mRNA, no? Removal of sequences. Leader sequences and introns that are not really going to contribute anything in the function of the transcript is considered as a post-transcriptional modification. Also, the addition of a CCA hydroxyl end at the 3' portion of the tRNA, that's also considered as a post-transcript modification. So, first, cleavage muna. Let's take a look at the cleavage of the leader sequence and some introns. So, here's your DNA template. This is serving as the leader sequence. This is serving as an intron that is not needed. So, primary transcript will still possess those things. Okay? And that primary transcript serving as a tRNA precursor needs to undergo cleavage. So under the action of RNase enzymes, endonucleases, the leader sequence and the intron will be cleaved. And ligase will, of course, fix the connection, no? Between elements of the primary tRNA precursor that needs to be put together. Kasi syempre, mawawala yung intron na yan eh. So it has to be, that gap needs to be closed. And that's via the action of ligase. Now, another element or another post-transcript edit is the addition of the CCA hydroxyl end via the action of tRNA nucleotidyl transferases. So, in the presence of energy, okay? So at the 3' end will have a CCA hydroxyl end later on. Magkakaroon ng CCA bases here, right? Also, we have base modifications. These are also considered as post-transcriptional modifications. Methylations, addition of a methyl group in our nitrogenous bases. Okay, we also have reduction, the addition of hydrogen atoms. Okay, transversion. So yung ating pyrimidine replaced with the purine or vice versa. And we also have deamination, removal of the amine group from our nitrogenous base. So these base modifications, they are also considered as post-transcriptional events, no? Post-transcriptional edits. Now, our last topic will be modifications in the ribosomal RNA. So the 45S transcript, which can be found in the nucleus, is actually the precursor to the 3 different types of ribosomal RNAs. the matured ribosomal RNA will be assembled along with ribosomal proteins to form the ribosomal complexes that are exported via the nuclear pore complex to the cytoplasmic or the cytosolic space. Okay, so 45S again will give rise to those matured elements. Okay. Now, in addition to our discussions, we're actually done. No? I just want to include one additional material, the ribozyme. Okay, so ribozymes are special enzymes. Okay, that are found to be capable of or a special RNA stretches that are capable of self-splicing. Okay, so this catalytic RNA is capable of self-splicing. And what do you call this? Ano siya, no? Yung kanyang catalytic domain and kanyang substrate where the catalytic domain acts upon is located in the same molecule, forming a hammerhead structure. So here you can see the hammerhead structure of the ribozyme. Now, the ribozyme is very important. The discovery of it as a very active molecule, a self-splicing molecule, because I don't know, it supported the idea of the RNA first world. No, meron kasi tayong conflict, not really conflicting, but alternative hypothesis on the evolution of genomic and genetic systems in bacteria and also in eukaryotes. So yung mga naniniwala doon sa RNA first world, yan, one of their evidence is the existence of ribozymes that are capable of self-splicing. No? We've discovered a lot from ribozymes, no? That they've been used for artificial purposes, no? Na kaya sila, no? They can act on certain substrates designed or that are designed to be that are artificially designed to be acted upon by them. Okay. That's it. That's the last slide for our lecture on transcription. So I hope we're very clear with RNA transcription, not only in the prokaryotes, but also in eukaryotes. And we also covered a very nice introductory lecture on post-transcriptional modification. Okay? Make sure that you have covered or reviewed our curated material, recorded lecture in replication before you listen to this recorded lecture. Okay? So the next video I will upload is on translation.